Hi and welcome back to another A-level biology video. In our last video we looked at the structure of HIV and how it replicates. In this one we're going to look at how we test for HIV. The type of test that we're looking at today is called an ELISA or an ELISA test. That stands for Enzyme-Linked Immunosorbent Assay. It works on the principle that a person who's been infected with HIV will have antibodies against HIV in their blood.
First, a layer of ready-made HIV antigens is bound to the bottom of a well in a plastic tray. Next, a sample of the patient's blood plasma is added to the well. If there are any anti-HIV antibodies in the plasma, they will bind to the antigens at the bottom of the well. The well is then washed out, removing everything except the antibodies bound to the antigens at the base of the well. The antibodies are too small to see, so at this stage we don't know whether they're there or not. So we add another solution. This solution is full of another type of synthetically produced antibody that has an enzyme attached to it. This secondary antibody has a binding site for anti-HIV antibodies. So if there are any present, stuck to the bottom of the well, the secondary antibody will bind to them. Next, we wash the well out again. The antibodies with the enzymes attached will only stay stuck at the base of the well if there was anti-HIV antibody present there. If not, all the enzyme will be washed out. Now for the moment of truth. The last thing that's added is a solution that undergoes a colour change in the presence of the enzyme that was attached to the secondary antibody. So, a colour change will be observed only if the anti-HIV antibodies were present in the patient's plasma. A colour change indicates a positive result for the HIV test. So let's recap really quickly. If a person has HIV, they will have anti-HIV antibodies in their blood. HIV antigens are bound to the bottom of a well. The patient's plasma is added. If HIV antibodies are present, they will bind at the bottom of the well. The well is washed out for the first time. A secondary antibody bound to an enzyme is added. These secondary antibodies will stick to the HIV antibodies if they're present. The well is washed out a second time. Finally, a solution is added which undergoes a colour change if the secondary antibody with the enzyme is present, indicating a positive result for HIV.
So that's it for this video. Please like and subscribe if you want, and follow the link in the description to find worksheets to check your knowledge on this topic. I'll see you in another video. Thanks for watching.